My name is Ekaterina Alexeyeva, and I'm from LEXOGEN, and it's my pleasure to moderate this demo today. It will take about 30 minutes and will be followed with a questions and answer session. Please feel free to write your questions in the chat on the right side of your screen during the talk and the QA session, and they will be answered after the talk. Several of technical specialists from LEXOGEN and Bluebee are present here today at this webinar, and they are happy to help you. I have seen in the registration form answers that there are some of the attendees who are still not using QuantSeq, and therefore, for the beginning, I would like to shortly introduce it. QuantSeq is a LEXOGEN answer for expression profiling sequencing. There are two types of library prep for RNA sequencing. In whole transcriptome sequencing approach, libraries are prepared from many fragments along the whole length of the transcript. The transcripts concentrations are calculated here through SPKM values, which are probabilistic estimates of isoform abundances. In expression profiling approach, libraries are prepared from just one fragment per transcript, and therefore data analysis is based on counting of transcripts without need for SPKM values. QuantSeq is a dedicated library prep protocol for expression profiling, which means that libraries are prepared from one fragment per transcript, particularly next to poly A tail. The whole workflow just takes 4.5 hours to complete. This protocol has many benefits. QuantSeq is the best sequencing-based alternative to microarrays. It can be used for genome-wide analysis of genome expression. It has exceptional reproducibility and sensitivity for the low abundance transcript detection. The price per sample goes down to $20 US. QuantSeq works with as less as 100 picograms of total RNA input and can be used with low quality and FFP samples. QuantSeq allows multiplexing of up to 96 samples in one lane of your sequencer. Data analysis is simple and can be performed even without bioinformatics experience, and this is in fact the focus of our talk today. QuantSeq data analysis pipeline has three major steps: quality control, alignment, and gene counting. All of these steps have been incorporated onto the Bluebee platform, providing a QuantSeq user a free, very fast, and efficient data analysis tool. You do not need to have a bioinformatics experience anymore in order to do QuantSeq data analysis. Standard Bluebee setup includes read streaming with BB-Duke, alignment with a star, and counting with HTC count. Every customer of QuantSeq receives a code for a free data analysis on the Bluebee platform. And now I would like to pass the word to Valerie from Bluebee, who will continue with the demo and more detailed introduction of the platform itself.
Thank you, Ekaterina, and also on behalf of Bluebee, welcome to everybody to this webinar. Before we go to the demo, I would like to give you a little bit of background information on what the Bluebee genomics platform is about, and then we'll focus on how you can use the Bluebee platform in conjunction with your LEXOGEN QuantSeq kit. So the Bluebee platform is an accelerated private cloud platform for genomics data analysis. So the platform is highly secured and allows you to very easily start analyzing your genomics data, which could be RNA-Seq data, like here in the case of the collaboration we have with LEXOGEN, but it could also be whole genome, exome sequencing, targeted panels, etc. As it's a cloud platform, of course, everything that has to do with security is extremely important, and we'll come to that in the next slide. The platform is also very well set up for managed sharing and collaboration. So for those people amongst you that would need to share the results of the outcome of your data analysis of your QuantSeq kit with collaborators within your organization or outside your organization, I will be able to show you in the demo how you can do that. What's really important from the point of view from Bluebee is that the scientific validation of our pipelines is essential. So we have in our platform a number of ready-to-use reference pipelines, but we have also a service where we lift over clients' existing pipelines onto the platform, and after scientific validation, you can start running these platforms and these pipelines on our platform. So this is exactly what happened with LEXOGEN. So LEXOGEN has developed a pipeline. We have implemented it on the platform for you, and it is now becoming a ready-to-use reference pipeline in conjunction with the kits. So the convenience is a very important aspect. As Ekaterina told you, there's no bioinformatics knowledge required. You can start using the pre-configured pipeline right away. There's a very limited setup in terms of user data and upload configuration, and there is a project ready to use for you that has been implemented by the LEXOGEN team. Important also is that for the QuantSeq users, the data analysis on the Bluebee platform is included in the price of the kit. Obviously, security is a very important aspect. This is why we work in highly secured compute centers. We have a global rollout over 22 different data centers, meaning that we can always make sure that the data is protected according to local regulatory requirements. The platform itself is ISO certified, HIPAA certified for the US, and is compliant with the EU model clauses for data privacy. So all the customers' data are strictly segregated and encrypted at all times. This is really important. Also from a Bluebee perspective, we will never access your data. We will never use or share your data or your pipelines with any third parties. So before we go to the demo, a little schema that would explain how it works. So on the right, you see the Bluebee genomics platform, which is in the cloud. And then here on the left-hand side, you would typically have your sequencer. The sequencer would output data, typically FastQ data, to a computer or to a network location. And this is where a little tool, which is called the Bluebee Service Connector, will automatically pick up the data, encrypt the data, and send it automatically to the Bluebee platform. You as a user can access the platform via the web GUI, and at all times you can see what the result is of your data analysis, or also how your analysis is progressing. For big volume users, we also allow API-based integration to automate everything end-to-end so that no user access is required. We offer three kinds of setups. We have a number of off-the-shelf pipelines that are available on the platform and that are ready to use. So the LEXOGEN QuantSeq Pipeline is one of these. For power users, we also offer the possibility to create and configure your own pipelines. And a third option, if you would already have an in-house pipeline, we have a service where we can do what we call a liftover of your existing pipeline, implement it on the platform and add it to your library of platforms, in which case, obviously, these pipelines would be only visible to you. So that's it from an introduction perspective. So the idea is that we would go now to a demo so that I can show you how everything works on a day-to-day basis. So when you want to start using the Bluebee platform, the first thing that we would recommend you to do would be to go to www.bluebee.com/QuantSeq. On this page, you can read a little bit more about the partnership. You also have 4 videos that will explain you how to register to the platform, how to connect your sequencer, upload your data, and retrieve your results. So if you really want to have a slower version of what we are doing in the demo today, I would recommend you to look at these 4 videos. And there's also a user guide that you can download. So once you've figured everything out, you can click on Login to the platform. And this is your screen that you will initially see. Obviously, the first time that you come onto the screen, you would not have a username and a password yet, so you would need to register to the platform. In order to register to the platform, it's important that you have the kit code that comes with your LEXOGEN QuantSeq kit. So if I click on register, there's first of all a CAPTCHA. And then here you can fill out your kit code. So let me take a kit code. And you continue. This will bring you to a screen where you can fill out your company account, where you can choose your username and your password. You give us your email address, your personal information, you accept the terms and conditions, and then you're registered to the platform. Once you've filled out that screen, there's only one extra step, which would be to go to your email to validate your email address, and then automatically you will be able to log in to the platform. So let me go back and log into the platform. So this is what you would see the first time that you log into our platform. So obviously no runs have been executed yet because it's the first time that you're accessing our platform. You will see here under the kit codes that you have purchased a kit with 96 samples, and at the moment 96 of those 96 samples are still available for you as credits, as allowed slots to process on the platform. The project is the most important part of what you will use. So here you will find a predefined project with some explanation that contains the pipeline for the QuantSeq forward. So if you look here at the pipeline, you've been able to see the different steps of the pipeline as they have been defined by LEXOGEN. If you look at the data, obviously no data has been uploaded yet because you're a new user to the platform. As you remember from my slides, the first thing that you will need to do is install that little tool which is called the Bluebee service connector. So that's the initial setup. You will have to do that only once, and this is how you will let the Bluebee platform know where to pick up the data from your internal network or from your sequencer and upload it automatically to the platform. So for this, you click on connectivity, you request a new connector, you enter a name, you define the operating system, and you will download the little tool. Here you have your license key, and the only thing that you'll have to do is go to a wizard that will, with a few clicks, allow you to install the connector. I'm not going to do that now because I already have a connector on my laptop, obviously. So this is then how it will look once it's installed. As you see it here, it's still orange, but after you have activated it with the kit code, it will turn to green, and you will be able to progress with the upload of the data. So once your connector is active, the only thing that you have to do is to let the system know where to find your files. So if I click on Add New, I can here select a name, write the file folder, where the data will be found, and define which input format. In this case, it's going to be a FASTQ file. And then you can do the same with the download data. Downloading of the data is downloading of the results. Obviously, you can always visualize the results in the platform, but if you want to have them automatically sent back to you, in the same way as you configured an upload rule, you can also configure a download rule. Let me now switch to a user who has a little bit more of data in the platform already. Just a second. So this is the same lab, but let's fast forward a little bit in time. 12 runs have been executed yet. So what do I see in the platform? I see the same description. In the connectivity, I see that there's 2 connectors active. This is important for you to know. You can make as many connectors as you want. So if you have different sequencers or you're working with different core facilities that would sequence the data for you, you can have as many connectors as you want. And for each of the connector, you define upload and download rules. Once these are installed, you don't have to do anything. As soon as data has been put into that folder C data upload, automatically the system will pick it up, encrypt it, and in a secure way transfer it to the platform. So you will find the data here, and here you see the FASTQ files that already have been uploaded. So from here on, you can select the FASTQ files that you want to process, and you can start a new run based on the pipeline that is available for you. And the run will start, and you will see here that the data is being processed. Once the data is processed, it will appear here. If you look at the runs, you see a history of all the runs that have been executed for you, and you can also see here the date, and you have behind in the back you have a full audit log on everything that has happened in the platform. If you now double click, you can see at the bottom the results of the data analysis. And this result of the data analysis you can view as I am doing here on the screen, but if you have defined a download rule, it will be automatically sent back to you without any manual intervention. Now, I have shown you how you can install a connector. I've shown you how, if a connector is installed, you can manually start a run based on data that has been uploaded. But if you use this on a regular basis, it would make sense to automate everything, and this can be done here in the connector. So if I go back to my upload rule, I can automatically here select the pipeline, and this means that no manual intervention will be needed anymore. So as soon as the data is available, automatically the data will upload, automatically the pipeline will execute, and automatically the results will be made available to you, either via the user interface or by downloading the data automatically. This is from an operations perspective how things would work. There's a few little other things that I'm able to show you. First of all, if you are in trouble for some reason, you can always create a support ticket, and you will get in touch with our help desk. So you let us know what your problem is, and immediately and also through the same user interface, our help desk will answer your question. A last thing that I would like to show you is what we call the permissions. So you have now pipelines in your project, you have now data that you have processed, and here in the permissions you are able to invite other people. So you can just add an email address, and you can invite me. I will receive an email and I will be able to to join your project. You can invite people, and once they have accepted, you can define their user rights. There's two possibilities. Either you give them view-only access if they're only able to see the results, or if you want them to be able to sequencing data themselves, you have to give them contributor access. But that would mean, obviously, that they would be able to use the slots that you have on your QuantSeq kit. This is now all based on the first kit that you have that you have uploaded, and you can at any point in time add an extra kit. So this is now the kit code. As you see, I've been processing 4 more samples, so I have 80 slots yet left. If at a certain point in time I buy a new kit, I don't have to go through the whole process anymore. I can just add the kit here, and then I have 96 or 192 or whatever kit you bought, more samples available for you to process in the platform. So this concludes it as a demo from what has been made available to you as part of your LEXOGEN kit. If you're using this platform and at a certain point in time you would like to use it for extra data, we can always open up your account. So this is how a Bluebee platform then would look like. You would see here that you have more options in terms of setting up your own project, and you can create projects which are exome or whole genome sequencing. You can set up collaboration projects, but this would obviously be part of another webinar if you would like to see how that part would work. So that concludes it a little bit of my part from a demo perspective. So I'm happy to take any questions, and so there's Yekaterina.
Thank you very much, Valerie. We have some questions. And the first question is, in which format should I apply the data? And I would like to ask our bioinformatics expert, Michael, to take this question.
Yeah, hi. Yes, Michael. The upload file format is usually FASTQ or zipped FASTQ files. So which are the, I think, most common formats for sequencing data.
Mm-hmm. Thank you, Michael. And the second question is, how long does the analysis take? And yeah, can you please also comment on this?
Yeah, apparently it's how it depends on the size of your input data, but usually in our test cases we had 10 to 20 minutes per sample, and since it's running in parallel, you have, I would guess, within 45 minutes the whole lane analyzed.
Mm-hmm.
So far, output will be then the gene counts per sample, but we are working also on a differential expression analysis pipeline so far, and we will integrate that also, that you then also can specify your samples and then compute the differential expression on that.
Thank you. And the next question is whether Bluebee analysis also comes for QuantSeq reverse kit.
Yes, of course. There are two, or depending on what kit you are purchasing, the codes are so generated that you get then either forward or reverse codes, which are corresponding with the pipeline.
Thanks. Is it possible to adjust the analysis pipeline to include top hat and cufflinks?
It could be, but it will take longer. We wanted to have it that the pipeline is working for forward and reverse. Because if you run a forward experiment and afterwards a reverse experiment for QuantSeq, it's hard to compare then these two output if you want to use top hat. And with QuantSeq reverse, you cannot use top hat because of the missing junctions. It's complicated, but we tried it with the QuantSeq reverse with top hat 2, and it just crashed. And links you don't need at all, since with couplings you compute the SPKM values for your transcripts and then go back on gene level, and that's not necessary because you cannot distinguish the end sites so well that you see the transcript abundances in a way to get that proper gene count.
May I change the parameters? Sorry. May I change the parameters of the pipeline? Valerie? Okay, so I continue. May I change the parameters of the pipelines, for example, genome to align to annotation to be used with the ICC count?
So far, we have preset genome annotations for the pipelines. It's the ensemble annotation of human, mouse, rat, and pig. And you can select for these species, but you cannot change the parameter for HTC count or something like that. Of course, you can add a new species anytime there's the need for it.
Can I perform differential expression analysis? Sorry, Valerie, yes, please go for it.
Yeah, I would like to add to this. So the QuantSeq pipeline has been defined by LEXOGEN and is offered as explained as a predefined pipeline. If you would want to use the Bluebee platform for other pipelines or to create your own pipelines, this is also something that is possible. And there you can choose, and you can choose your tools and you can change your parameters. But this would obviously then be something extra. It would not be included in the price of your kit. But that's certainly something that can be discussed.
Thank you for this additional comment. So the next question, I think it also goes to Michael. Can I perform differential expression analysis similar to CAT-B, for example?
You get gene counts, and you can perform any analysis which is suitable for gene count analysis, such as HR, DE-Seq2, and there are a plethora of different R packages to do this. CAT-diff is again on a certain model which has a coverage model for transcripts and which will not work with QuantSeq.
Thank you, Michael. Jose is asking which are the default parameters of the pipeline, which genome and which annotation used. I think you already answered this question, and but there is additional part of the question, if there is any reason to use a particular annotation.
So we had different testings on the annotation. So usually the RefSeq annotation, which is also quite popular, the genes are usually too short there. So the annotation is very condensed and has not so many different isoforms as in Ensemble. And usually also the 3' UTR is not so well annotated in RefSeq as in Ensemble. That's why we chose the Ensemble annotation. And yeah.
Okay. Is there an official minimal number of reads supported for the analysis?
We had it once in our application note, and I think 5 million is great, but you can go down to 1 million, and you can still have a good result in your differential gene expression analysis.
Julianne is asking, 3' sequencing being different from classical whole mRNA sequencing, do you use a special gene annotation for 3' sequencing?
As I said, for the ensemble annotation, it works fine. We had customers, I think it's also on our webpage, the webinar, our first webinar, where they sequenced chicken, and there they needed to extend the existing annotation. So they extended the 3' UTR downstream to, I don't know, a couple of kilobases or so. So it's, there was the annotation not so well. But for mouse, human, and rat, it's so well annotated in ensemble that you don't need extra definitions. Yeah.
Okay. I have read that one can do soft streaming using star with some parameters. Is there any reason to use hard streaming with BB-Duq? Is it possible to use Genomics or other streaming software? Which quality parameter is used with STC count?
So of course, yeah, you can do a soft clipping with star. We did a couple of experiments that, although setting soft clipping on, you get better results trimming with BB-Duq before. So we trim mainly the adapter contaminations. So you're reading into the adapter, and sometimes for QuantSeq forward, for instance, you run first through the poly A primer part, and then into the adapter sequence. So we also control for this poly A read-through. And of course, you can use other software like Trimomatic, TrimGalore, even the old FastX toolkit trimmers can work with it. But in terms of speed, this BB-Duq from the BB-Map suite is most suitable here. And the quality parameter for HTC count, as far as I remember, we used a minimum mapping quality of 10.
Maybe you have mentioned this before. If I use multiple barcodes corresponding to one sample, how can I specifically define this in the analysis settings?
So far, you cannot. You can either run the pipelines for the replicate and afterwards combine it. That's, for instance, possible for if you want to do differential expression analysis afterwards, to have a look at the variance which is needed to model for that. Or you can pool your data beforehand using, for instance, a simple cats command on the command line.
Thank you, Michael. In fact, this was the last question we had so far. Are there any other questions you would like to ask now? If not, I want to say thank you to everyone for attending this webinar. And if you want to get more information about QuantSeq and about the Bluebee platform and our QuantSeq pipeline on this platform, please feel free to always contact us, and we are happy to help you.
Thank you very much, also from Bluebee side, and thank you for Yekaterina for hosting this.
Thank you. Goodbye, everyone.
Bye-bye.